Good evening, everyone. Dear participants, we'll wait for another few more minutes for other participants to get joined. Um, Myself, Prasannaswamy, Luminol Biopharma. Let's give a brief introduction about our company. Luminol Biopharma is an integrated biopharmaceutical company established in Vietnam, the healthcare specialty and biosimilar medicine of socio-economics focus with our own technology and product development. So our R&D has an analytical and preclinical clinical development services. The company also offers the fledgling support to the academic towards developing the future of the company. So here today we have the basic introductions to the bioinformatics. So normally we know the bioinformatics. So we cover those topics on this today. What is bioinformatics and history about the bioinformatics, and the area where we can currently establishing and future development will be there for the scope of bioinformatics, and some molecular facts uh, linking to the bioinformatics and typical problem and issues we faces while we do the bioinformatics analysis and some application about it. So what is bioinformatics? We generally the statistics, biology, and the program and databases which all forms into the union sectors will have the bioinformatics sections. So here this integrated discipline of the all the sectors, including the biology, computer science, and information technology, and it has a role to play the statistical and computing methods that aim to solve the biological problems. So, for based on the DNA and amino acid sequences related to the informations. So, the amino acids and the DNA, so DNA and the nucleic acids, majorly we know that the sequence adenine, guanine, cytosine, thymine, and uracil. So these codons are involved in the basic the codon that develops the informatic structure. So that amino acids, we know that three letters formations and single letters were coded to the bioinformatics systems. So when you get the leucine, lysine, glycine, phenylalanine, and other proline materials respectively. So history, we know that Margaret Day of in the year of 1972, the first bioinformatician developed the biological database on the protein identifications resources. So then the team involved to develop the protein families and superfamilies based on the sequence similarity. The sequence alignment was introduced as well respectively and observed in the sequence of a group that where the protein can be closely monitored. And there are several huge protein banks as well as known today, Swissport and PAR International and other etc. So normally this database was established in the year of 1979. Currently there are several powerful databases like GeneBank, EMBL, DBJ, and other. And we have developed the soft tools like Fast Blast and Clusterwall, and these are the tools we are having, and other online tools also there to find the sequence of the DNA or the protein or alignment of the sequences we do. So these, what I said before, these 20 amino acids are common in living organisms where we can see. So these are the three letters generated and as well as the one letter code also involved in that. So this evolution, this helps in the evolution of the reconstruction process. So we can see the brief history of the bioinformatics on the evolutionary reconstruction, where there are three organisms as shown like A, B, and the C. So this process that involves a small or cumulative changes in the DNA that eventually leads to the formation of a novel species. So where you can see that, for example, in the organism A, that S represents the leucine, using the one second. So here you can see that evolution where an organism A to B and organism A to C. So W where it's reconstructed to the Y where this shows the leucine to the tryptophan to the tyrosine, and the other you can see WL2R, it shows that leucine to the arginine. So this evolutionary shows that DNA eventually leads to the formation of the novel species in this phylogenetic relationship. So next we can see the other important steps, the sequence retrieval method. So these are developed in the years of 70s, between the 70s and 80s. There is a sequence retrieval method, and in the year of 1980, they developed the sequence of principal sequence of the alignment and the prediction of RNA and secondary structure. So then in the year of 1980, the predictions on protein secondary structure and three-dimensional was aligned between the 80s and 90s. And as said, the FASTA BLAST method of database research was introduced in the 90s, and gene prediction and the complete genome sequences was in the late 2000 we have been determined. So these are the small brief history of the bioinformatics where we have been started, and stepwise there is a growth in the field of bioinformatics. So let me see the area, the current futures and development, where we can see mainly the applications of applications of molecular biology and genetics and phylogenetics and evolutionary science, and where in the field of medicine, agriculture, and eco management, and so we can nowadays we can find that it is more applicable in the area of pharmaceutical and microbiological industries too, and shows a huge growth in that area. So, all the bioinformatics, the advanced levels to the computational biology. So this computational biology is it's a conventional evolution population of the theoretical and the classical biological science, primarily concerned with the population studies and the theoretical analysis of the biological sequences rather than cellular or in the molecular level would be helpful. And in the medical informatics, the applications and computing the method to uh, improve the medical data, and we know the medical coding nowadays has a huge market in the to get the data from the doctor side, and they will convert to the algorithm part, and which is feed in the database as an medical code. Medical coding is a tool used as the major factor. So generally concerned with how the data is manipulated and how the data is itself registered to the program. So then another area which is cheminformatics, where we can know the both chemical and biological technologies for the drug design and development. So normally nowadays this area also is huge in the pharmaceutical and pharmacogenomics area to develop the drug and their development and designs involves. So, and especially now very enlarged area of genomics and proteomics, there is an comparison of genome to a single species or the multiple species, where you can analyze the sequence analysis, which the genome set to all the possessed by an organisms. So where you can exist the genome are completely sequenced, but it is very primitive state. Anyway, it's how the sequencing also have been made, and we have find all the sequences from the Sanchez sequencing to the nanopore. The technology develops all based on the biological, and the technology involves the bioinformatics system. The programs are feeded to analyze the sequences of the pipeline of every organisms or so infectious disease, the anywhere we can apply that things. So proteomics as well, the genome is expressed in the protein and flow. How these proteins functions where in fact we can be analyzed, and concern actual states of the cells and the potential state of the which cannot be described by the genome were also be analyzed in the proteomics side of the bioinformatics or computational biology concepts involved. So as said, for the pharmacogenomics, the applications were to test the target of the drug or the potential of the drug can be analyzed, studying the gene expression patterns in a tumor or any infectious diseases or any other disorders can be to detect for the diagnostic purpose. And the genomic methods are determined causes variation in individual response to the treatment of drugs involved. So to identify the targets, for example, searching the entire genome for the potential drug receptors where the expressions can be studied using this. So variance is responsible for the drugs can also be determined using the pharmacogenomics, which the help of the bioinformaticians. So this shows the central dogma of molecular biology, which all biotechnologists commonly know. So how it happens, a single strand of DNA to the complementary DNA, which replicates, and then your complementary DNA is transcripted to the mRNA sections, and the translations would help for the protein identification of the single. So what are the amino acids presence inside it. So this is the basic concepts which helps to develop the um, bioinformaticians for their all sequence analysis or any protein interactions could be done. All the bioinformaticians are done using this profile. So what are these are types of issues. The types of issues what we face: the collection and retrieval of data, the alignment method. So the sequencing of DNA and protein, and the database what we submitted, some could be difficult to get appropriate your the DNA for the sequence of DNA. We would need the complementary sequences. So sometimes there are some issues we can face in the alignment of data and methods. So next, the format we use for development of the sequences. Again, the files like BAM or the BIP files, what we use to be determined based on the concepts and the pipelines to be analyzed based on the development of the sequence formats. And conversion of one sequence format to another is also the time that which we need to concentrate more to study the DNA sequences and other analysis. So development and retrieval alignment methods, which also more is a problematic area to determine the alignment, the mismatch or matches could be to be solved in the SNP polymorphism and where the overlapping occurs. So then the prediction, reconstruction and classification, as we are earlier seen, where the classification of protein and genes or analysis of the genomes or any reconstruction of phylogeny, it's a work where to see the promoters and other functional sites. And now commonly we used this primer they're designing based on this all technology by the biological data is involved in the construction of the primer. This should be there should be a classification and other things to be did. The prediction of the secondary and the tertiary structures of RNAs and proteins of other. So we can see the few applications. We know the biobank facilities and other. So this is the biorepository which stores the human biological sample data. We know that, and we have been coded even though in the recent times we know we are once the baby born we are collecting the stem cells for their storation for further any for their health issues purpose for the future. So all this we have to code some data and code some barcodes based on the concepts, the cells which we have been obtained. So since the late 1990, the keys we can see the supporting of many types of research based on the genomics. We need a human genome project which has been completed in the market of, and now they have taken the challenges of exomes to calculate the sequences and analyze the sequences more in that. And some in the drug discovery and development of drug also, as the industry shifts to more personalized medicine, where the single person has genetic has been analyzed to give the drug for their response, where their body responds to to diagnose their disorders. So this need of the high quality, well maintained biospecimens and intensifies in the market of these recent. In the Western countries, we see more applications based on the pharmacogenomics, where they study the full genetic or genetic analysis of the person to even to describe the medicine for even small fever or any stomach upset or anything. Where here they won't try for the drugs, so which it works or it may not works like that. They have complete sequence analysis and they find how the drugs to be targeted in the site of where they need to be diagnosed their disorder. So in the case of veterinary science or animal health, divided to the protection of animals and companion animals. The animal health market covers the diagnostics and the medicine for the veterinary animals and the farm animals and pets. Growing demand for the animal protein, as well as we know the strong need of companion and health care to be provided. Still, there is a question mark in the applications, but this scenario will develop the more appropriate solution for the animal health, and the veterinary science people will get this analysis as well. So here we can see the growth of the gene bank from the 1982, 2006. There is more sequences developed rather than the annotations. So this bioinformatics has a drastical growth than what the wet lab people do. Before that, they required help from the dry lab, what is the bioinformaticians or biotechnology, to give their inputs on where they can stand their research part to develop efficiently without going out of any losses in the loss or any losing part of the research scenario. So which helps which the researchers are more depends on the bioinformaticians to give the appropriate things. How the things works on the development of the research. So again, we can see the pharmacogenomics already what we discussed. So all drugs works on all patients. It's not possible. So what I said earlier, some good drugs causes death in some patients even because based on their genetics, based on the single person or their metabolisms. So by doing a gene analysis before the treatments, the offensive drugs can be avoided. So the sequencing will be analyzed based on the sequences, and the doctor can suggest the patients what kind of drug can be taken. For example, there is we know most commonly the diabetics. The diabetics the world follows the two type of the drug. One is like metformin based analysis, and another one is sulfonylurea drugs. So mostly the sulfonylurea drugs is cheaper than when comparing to the metformin. So normally the doctors uh, from the from low side they used to give the drug on a trial basis here. So just they will check whether the drug is working or not. So after that, once they got 3 months of medication, drug therapy, the patient still their insulin was not regulated properly. So again they will go for an treatment. So by the time the doctor will take a call of changing the medicine to the another formula like metformin or some another. So again he will be diagnosed. So the drug taken is not cured the disorder, and it means that it's a settled down as a it is settled down, and it will cause the side effects in the future. So the must thing is to analyze their sequences, their DNA, their gene, to give the drug therapy. So which causes to avoid any side effects or any drastic thing of like the death. So that's the pharmacogenomics where these applications involved for the customized and treatments. And we know the gene therapy for the replacement or the supply the defectives or missing gene. So example, if in the insulin, the factor 8 or the hemophilia, which The misplaces, the gene or overlapping or mismatching can be analyzed based on the gene therapy. So the gene therapy also plays the important role based on the bioinformaticians or computational biology. So that and diagnostics of diseases. So we can identify the gene which causes for the disease or disorder helps to detect in the early stage. You know the Huntington disease. The Huntington disease. The Huntington disease normally it causes the mental depression and it affects in the brain. So this can be detected using our being as sequencing being the sequencing, we identify the cracks and where it happens to damage in the gene sequences or anything can be altered using the gene therapy. It's a future diagnosis to it's a future diagnostics helps in further treatment of medical science. So, as I said, the symptoms is mental disturbance or personality changes and intellectual impairment. But we seen about the Huntington. The gene responsible for disease has been identified based on this. So the drug design. So normally it takes a 15 years and lot of investments in for to design a drugs. Bioinformatics helps in that way there where they can reduce the time and cost. So the process and discovery can be earlier when compared to the other for any like what we have been faced in the corona during the period of corona. Very short time the vaccination has been developed and saved a lot and lots of people's life. So drug design again, the bioinformatics part is more involved in the drug discovery and drug design. So it helps to reduce the time and the other cost factors. So for the targetic identification, identifying the molecule on which one see the drugs which will be the blind to the target. So we have to say this bioinformaticians helps the drug target region. So where there is a pathway involved in the system, whether maybe in the endoplasmic reticulum or any other metabolic sites where can be with targeted drugs should be um, get into that area to resolve the to start the diagnostic purposes. So identifying the molecules for the survival purpose, the again the site of bioinformatics is used to detect the path away. So the germs will not be able to interact with the targets, and the proteins are where the proteins find to be most common target where they can get attained to that. For example, so HIV produces the protease, which is a protein, and which in turns to eat other proteins. So this may be the protease has an active site where it binds to the other molecules. So the drug involved in that will go and bind with the active sites. So easily it can restrict the restrict the sites of other germs or protein involved in that area. So expectations for the students in this course. So basically, we understand the general principle of the molecular biology involved in the bioinformatics. So some mathematical and computer science background will apply to in the area. So we have to focus on all the computational methods and understanding the general analysis used in the bioinformatics for the description of the algorithm concepts and methodology will not be the core element, even though the basic things we need to be addressed in the part of developing the computational biology area. The core requirement to understand the foundation of bioinformatics and the application approaches, where you get in the different area where we see the diagnostic part, pharmacogenomics, drug therapy, gene therapy. So these are the areas where we focused to develop the bioinformaticians to find a solution in that area. So generally, we are also providing our Sri Sai Institute for the containing dedicated training, workshops and other things. So we are also planned our workshops in the bioinformatics using the tools, online tools and other offline tools which we have been with us. So we are intent to give our workshop. It will be notified when we plan the durations and other. So we have the department like molecular biology, bioanalytical, and bio in the small molecules and large molecules, and we have the facilities of instrumentation like HPLC, LCMS, and other things. You can contact through the details, and we have the opportunity to give the college students an industrial visit purpose, which this shows that previously we had a batch of 48 students who have been take part in the industrial visit in our organizations too. So you can note the training purpose. You can note the contact, the EA calls or the email IDs for the industrial training and visits. So we provide a webinar on the next section for the introduction to the basic immunology by our honorable speaker, Dr. Satyabhama. He is a senior scientist in our lab. So next section will be planned accordingly and will be notifying through the mails and other social sectors. And we are kindly request you to fill the feedback form which will be shown in the comment sections. Thank you for the wonderful time you have. After filling your feedback form, you will receiving your certificate, and any questions can be given in your comment box, can be replied. The protease are the efficient the executioner of the common chemical reaction that hydrolyze the peptide bonds, which most proteolyte enzyme cleavage has alpha peptide bonds between naturally occurring amino acids. So, any other questions? So any more questions are there, or shall we wind up the classes? So the gene bank is the most complete collection of the annotations, nucleic acid sequences. We know the data for the nucleic acid sequence data for almost every organisms. So it includes all the genomic information of the DNA, complementary DNA, mRNA, and high throughputs. We have the raw sequences for the sequence polymorphisms. So before the NGS training, we have a lot of basic platform to be learned in the bioinformatics tools and files, uh, how to analyze the files and pipelines. So after that, we'll move on to the NGS. NGS, we have a separate topic for webinar to cover the from the wet lab part of the NGS and how the dry lab part has been analyzed and everything. So once you complete all your feedback, we'll be getting a certificate as soon as possible. So with this, we'll wind up our section. Thanks for all the participants who have participated in this. Thank you.